Hello, this is Bio 225 Genetics from Genes to Genomes with Dr. Lucy Abgarian, Chapter 5, Part 3. So far, we have talked about diploid systems. Diploid systems such as mice, rats, Drosophila, humans. In these systems, each individual represents one of four possible parental gametes. Those parental gametes are product of meiosis. Therefore, when we are talking about recombination or linkage or gene mapping, there are many possibilities. There are so many possibilities of combinations of the segregation or linkage that we have to examine a large population for being able to make a conclusion and do the final and the more accurate mapping. We have seen this already in our previous examples when thousands of examples were examined in order to come out with a probability, more accurate probability of final mapping. However, there are some organisms, such as some fungi, that can give us opportunity to analyze this differently, as they can pass all four gamma haploid products in one sac, which is called ascus. Furthermore, these haploid cells can grow into haploid individuals growing by mitosis. And the beauty of these systems is that the genotyping is very easy, as their phenotype is exactly their genotype. Here is a question for you. What is the difference between genotype and phenotype? So here is a refresher from last lecture. We talked about this slide already, where we have in the bottom the diploid cell system, which through meiosis goes to association of ascus, and then the germination process, and we can see on the left and on the right different haploids with phenotypes that are basically synonyms. And the nice thing about this system, as we mentioned already, is that in these systems the genotype is the phenotype is representing the genotype. So here is an example of the life cycle of Neurospora crassa. The difference is that this system goes through two meiosis processes, and then it goes through mitosis, and it generates 8 haploid cells. Okay, by now it should be clear for us that the phenotype of haploid fungi is direct representation of their genotype. So let's look at some mutation situations. So mutations in haploids sometimes can affect appearance of cells and ability to grow under certain conditions. So let's see an example of histidine 4 mutation. With histidine 4 mutation, because of it being a recessive, the haploid cells one haploid cell will grow. The ones they don't contain the mutation, the other haploid cell will not grow because it contains the mutation, and the diploids will still grow because it's a recessive gene. And the same situation is with the tryptophan 1 mutation, where one haploid containing the mutation will not grow. The one not containing the mutation will grow, and the diploids will grow because it's a recessive mutation. Probably you have already noticed that there is a different notation for recessive and dominant system. When we are dealing with recessive system, we are using small letters, and when we are dealing with a dominant system, we are using capital letters. Okay, let's look at this schematic example here now, where we have two haploid cells, A and alpha. A is here and alpha is here. Each one of them contains one recessive mutation and one dominant mutation. For A, histidine 4 is recessive and tryptophan 1 is dominant, and for alpha, histidine 4 is dominant and tryptophan 1 is recessive. When they come together and they create the diploid cell alpha, A alpha, then they carry these two, histidine 4 recessive, histidine 4 dominant, and tryptophan 1 dominant and tryptophan 1 recessive. So in this system, if you look, you have both dominant histidine 4 and tryptophan 1 genes. So this diploid cell is going to be fine and grow just perfectly because it doesn't need histidine or tryptophan presence for continuing to grow. However, the parental system is going to have issue to grow without tryptophan or histidine. Okay, let's go deep inside this example and talk about three kinds of tetrates that meiosis can generate. So one is parental ditype. So here is the same example, right? When we have recessive histidine 4, dominant tryptophan 1 as A, and we have histidine 4 dominant and tryptophan 1 recessive as alpha, and we are looking at them as a parental haploid cells and what's happening next. So at meiosis 1 stage, we see that histidine recessive histidine 4 recessives are coming together, tryptophan 1 dominants coming together, and then the histidine 4 dominants come together, and tryptophan 1 recessives come together. This is that situation. And then the meiosis 2, we are having this situation when we have histidine recessive, tryptophan dominant, and we have histidine dominant, tryptophan 1 recessive. And then also we are getting the final tetrates, which is histidine 4 recessive, tryptophan 1 dominant, and then we are having histidine 4 dominant and tryptophan 1 recessive. So, and this is our ascus, formation of ascus. Here is another kind of tetrate, non-parental ditype. So let's look at the difference, and the difference should be seen from the very beginning, meiosis 1 stage. And the difference is that when in previous case we were dealing with histidine 4 recessive, tryptophan 1 dominant, here we are dealing with histidine 4 recessive, tryptophan 1 recessive, and then the second one is the histidine 4 dominant and tryptophan 1 dominant. And then therefore meiosis 2 is also looking differently, and we are having histidine 4 recessive and tryptophan 1 recessive, and we are having histidine 4 dominant and tryptophan 1 dominant together, going together. And then therefore the tetrate is looking also like this, and our ascus is having this look here. And then finally, this is the third kind of the tetrate generated by meiosis. It's called tetratype. And so situation here is that during meiosis 1, we are observing crossover, recombination, and we already have talked about crossovers. And here, just to compare with the kind 1, the parental type, the difference here is that we are having, instead of recessive histidine, we are having dominant histidine 4, which is causing in meiosis 2 recessive histidine 4, and the recessive histidine 4 is being substituted here by dominant histidine 4, and then the rest is remaining the same as it would be in case number 1. However, the tetrate situation is changing here. You can see this is the difference compared to the first situation, and the same is here, and therefore the ascus looks like this. So here is the tetrate analysis of unlinked genes. So when the genes are unlinked, number of parental is almost equal to number of non-parental, and this is statistical data. So for parental 31, for non-parental 28%, or number of tetrates. And for the tetratype, the number is slightly higher, it's 41. However, in case of linked genes, the tetrate analysis looks different, of course, which is kind of logical to predict that parentals will be the majority for linked genes. And here we can see that for parentals it's 127, for non-parental it's 3, and for tetratype it's 70. So here let's look at 2 different cases where there is a crossing over and there is no crossing over, and there is a single crossover. In A case, there is no crossover taking place, and we can see that those are the parentals, and this is the final product, and we can see that we have parental dye types here. And in case of single crossover, here is the crossover taking place, and of course it will result in tetratype products. And here let's look even more complicated cases. So when we have crossovers between linked genes, more than just one crossover. So case C, when we have double crossover, and again that will result in parental dye type, and case D, when we have 3 strand crossover, then that will result in tetratype again. Case E here is another case of 3-strand crossover, which is causing into tetratype again, and the F case is 4-strand crossover, which is causing non-parental dye type. You can look more carefully to see how it goes from parental linked to all the way to the final product. This is the formula to calculate the recombination frequencies in tetrate analysis. And going back a couple slides, there was a table, figure 516, and if we put all the data from that table, and we can calculate the recombination frequency for that case, which will give us 19 MOP units. So here let's look and understand if we can show that recombination takes place at the 4-strand stage. So let's say it did not take at this stage, and then we will have AB here instead of AB plus, and same here, and also here we will have A plus P plus, which will mean that our final product will not look like this or this. So therefore, and you can do the detailed analysis on a paper by yourself just to make sure you are getting this. And that proves that the recombination is taking place here and not here. So here is an evidence for exception to the rule that recombination is reciprocal. So meiotic recombination almost always generates equal numbers of both kind of recombination progenies. The ratio will be 2 to 2, but however there are rare tetrates that will segregate with ratios such as 3:1, 1:3, 4:0, or 0:4. And we will talk about implication of this for molecular mechanisms of recombination when we are discussing chapter 6. On the beginning of the lecture, we talked about neurospora and how it creates 8 haploid cells, and so it undergoes meiosis 1 and then 2 as usual, but a single round of mitosis that occurs after second meiotic division. That's what is resulting in production of octad. Ascus is very narrow and spindle forms parallel to long axis, as you can see here. And two genetically identical ascospores are next to each other. An arrangement of chromatids can be interfered from position of ascospores. Just applying what we have learned already, now we can basically see the process from the beginning to end for the segregation patterns. So meiosis 1, and we have two different color genes here, and kind of it's just very straightforward process which is causing in for white and for black, and same situation is it can be like in this order, in white first, white bottom, black top, or black bottom, white top order. So here is the case when crossover is taking place, and based on what we have talked already, you can follow again the pattern from the beginning to the end, and you like just looking on the end product, we can always say where at which stage the crossover took place. So we will stop here for this part of the lecture, and we will continue from here on during next lecture. Before we go further, I would like you to review everything that we have done, because during next class there will be a quiz, there will be a summarizing of this topic with a little more deep analysis with formulas, and also there will be quiz that will cover everything that we have done until now.